Good afternoon and welcome to this special edition of Science Sundays. Science Sundays is one of the premier outreach events of the College of Arts and Sciences, which includes natural and mathematical sciences, social and behavioral sciences, and the arts and humanities. I'm John Meekum from Physics and Astronomy, and I'm the chair of the Science Sundays Committee, which is composed of people from across the college. We work together to make this event the wonderful thing it is, and we're grateful for your interest and attention today. Today, we're doing something special. We're partnering with the Center for RNA Biology, and therefore I'd like to introduce their director, Professor Karim Forsyth.
Thank you, John. So yes, on behalf of the Center for RNA Biology, I just want to welcome you to this event and tell you a little bit about the center. The center was founded became an official university center in 2012, and our initial director was Dan Schoenberg, for whom we are naming this lectureship, and Dan's in the front row here, and just wave, and he's gonna say a few words at the end. And so the RNA Center has grown since it started in 2012. We now have 45 labs throughout OSU's main campus and also Nationwide Children's Hospital doing research in all areas of RNA biology. And this includes, fundamental science, but also therapeutics, which is, of course, what you're here to hear about today. And in addition to our research mission, we have an education and outreach mission, such as this event. And in terms of our education mission, there are undergraduates, graduate students, and postdocs doing the research on RNA throughout the campus. And today, they're really excited to tell you about their research. And so after the lecture, there's going to be a poster session with some yummy refreshments, but we really want you to interact with our trainees, and I hope that you hear about the exciting RNA research that's happening here at Ohio State. And the posters that are being presented are also listed on the program here. So now I'd like to introduce Dr. Dawn Chandler, who has a research lab at Nationwide Children's Hospital in the area of RNA therapeutics, and she's going to be introducing our distinguished speaker.
Thank you, Corinne.
Thank you.
Thank you all again for being here. It's my pleasure today to introduce Dr. Adrian Krainer. He is a distinguished professor, a St. Giles professor at Cold Spring Harbor Labs in Long Island, New York. And I've known Adrian since I was a graduate student, and I've known his work very well as a very basic scientist, understanding how RNA processing happens and how that occurs in cells at the very basic level. And it's been a great joy to watch him be able to translate that into therapies for children with neurological diseases. So it's been really wonderful to see that transition from a very basic science lab into a therapy that is now helping lots of people be able to walk and survive. It's been really wonderful. So I'm going to keep this short because I was asked to keep it short, but he has a very long list of accolades and prizes. He has won the 2021 Wolf Prize in Medicine and the Life Sciences Breakthrough Prize, along with Frank Rigo, who helped discover this antisense oligo process in medicine. And recently, as 4 days ago, he also won the Albany Prize, a Medical Center Prize in Medicine and Biomedical Research. So in addition to his academic work, Dr. Krainer serves on numerous scientific advisory boards and holds 13 patents. He will share today his cutting-edge research on antisense therapies for rare diseases, a field that is revolutionizing treatment options for previously untreatable conditions. And please join me in welcoming Adrian Krainer.
Thank you, Dawn, and it's really a pleasure to be here, and thanks to everyone for coming here on a beautiful Sunday afternoon to hear about science. So I've known Dan Schoenberg for, decades, and as a colleague in the RNA field, so it's a pleasure to give a lecture named in his honor. So let me get my laser pointer. I have an outline of what I want to talk to you about today. So the main thing is to describe a type of drug called an antisense oligonucleotide, or ASO, which was developed for a particular disease, spinal muscular atrophy. And I feel like bringing coals to Newcastle because a lot of research relevant to these areas is done here at OSU and Nationwide Children's Hospital, where there are prominent RNA researchers as well as clinicians working on this particular disease, and even a different therapy was developed here for this disease. So in order to explain this somewhat esoteric topic, I have to introduce quite a few concepts, and I'm going to try to avoid using jargon as much as possible, or I will define the necessary concepts or terms. So in order to get into that, I have to go back to some basic principles about genes and gene expressions and DNA, RNA, and proteins to be able to explain what RNA splicing is, and the fact that our genes are composed of exons and introns, and I'll tell you what those are. And so that will allow me also to introduce antisense technology, ASOs, which is a type of drug, a fairly new type of drug. I'll tell you about the disease, SMA, and then I'll be able to describe how this particular drug was developed and what its mechanism of action is, and what impact it's having on patient lives. And this is just an example of a type of drug, so I hope to have some time to describe other applications and ongoing development of this type of drugs. So let's start with some very basic things, namely DNA, RNA, proteins, and their building blocks. And so DNA is this molecule here. It has a very famous double helix structure. So it's the molecule of inheritance. It carries the genetic information. And RNA, unlike DNA, has only one strand, but it also can carry or transmit this genetic information. And so both of these together are known as nucleic acids. Proteins are very different. They have different building blocks. And based on the order in which those building blocks are present, that dictates that the proteins can fold into specific conformations and then do things like promote reactions, like enzymatic reactions, or they can serve as structural components in the cell or in the body. And so the proteins do all the work in the cell. And so these are the building blocks. So DNA is made up of 4 deoxyribonucleotides, and the abbreviations for them are A, T, G, and C. And RNA also is composed of 4 building blocks, very related, A, U, G, and C. Whereas proteins are composed of 20 different amino acids, and those are the 3-letter abbreviations for all 20. So going back to DNA and the double helical structure, so this was described by Watson and Crick. This is a young Watson, this is Crick, in 1953. And this is the structure that they described. And it was really possibly the most important advance or discovery in the history of biology. So actually they earned the 1962 Nobel Prize, so just 9 years later. And so this is more of a diagram of the same structure. So this is a double helix. And what's important in it is those 4 building blocks that I described, which are shown here, the bases, they form base pairs. And so each rung of the ladder is a base pair. And so these are the chemical structures of those bases. And then RNA has very similar building blocks. And then the backbone is made of sugar and phosphate. And so the sugars for DNA and RNA are almost the same, except at this position and this position. And the phosphate is not shown, but it's what joins the sugars together along the backbone. So these 2 strands run in opposite directions. And what's really important, and it explains the structure and everything, is the fact that the building block A always pairs with T, and G always pairs with C. And so if we determine the order of the bases, these nucleotides, along one of the strands, it immediately tells us what the order of the bases is on the opposite strand because of these base pairing rules. And so when we talk about sequencing, It means that we're using techniques and methods to read exactly what are the bases at each position and what order they are present. And that information is crucial, is very important. So here's a cell with a nucleus and a cytoplasm, and the genes are in the nucleus and they're made up of DNA. And I'm only showing you a little piece of DNA, but in reality, each of our cells has 2 times 3 billion base pairs, and they're organized into chromosomes. And for each chromosome, one comes from the mother, one from the father. And so what is gene expression? Gene expression refers to the fact that even though each of our cells has the same set of genes, about 23,000, they're not all functioning at the same time. Some genes are functioning in all cells, and other genes function in, let's say, liver cells, but not in brain cells, and vice versa. So when I say functioning, it means that the information present in that gene is copied to make a messenger RNA molecule. So that is a process called transcription. So there's an enzyme that reads the DNA in that gene and makes a precise copy in the form of a messenger RNA. So that's transcription. Then that messenger RNA has to be exported to the cytoplasm of the cell, and then it's decoded there in a process called translation. So there's a machine in the cytoplasm of the cell called the ribosome. That's what a ribosome actually looks like. It's made up of some large RNAs and proteins. And then it uses these adapter molecules, transfer RNAs, and here's a particular transfer RNA, each of which has an amino acid at its end, and then another portion that is used to pair to that messenger RNA. So the ribosome moves along the messenger RNA. These adapter molecules recognize these words of 3 building blocks at a time to decide which amino acid is going to be incorporated in the growing protein chain. So the ribosome is an amazing machine because if you think about it, it's translating the language of DNA and RNA into the language of proteins. And to do that, it uses we call the genetic code. So it's this table. And this table was worked out in the 60s. It led to the 1968 Nobel Prize. So the words are of 3 letters, and because there's 4 kinds of letters, that means there are 64 words. And yet we only need to encode 20 amino acids. So there's redundancy. So for instance, you see here that the word UUU and UUC both encode the same amino acid, Phe, which is phenylalanine. There's also a word that means start here, and it also encodes a particular amino acid, methionine. And then there's 3 words that indicate stop here. So when the ribosome gets to that point, it stops. That's the end of the protein chain. And so those are 3 stop codons. Now, if a gene is mutated, meaning that one nucleotide in the gene becomes something else, you can have different kinds of mutations. A nonsense mutation is when a particular codon, for instance, UAE, what do I have? UAU changing to UAG. So instead of incorporating an amino acid, tyrosine, now the ribosome stops right there. So you would get a truncated protein. That's typically a severe mutation. A missense mutation is if, for instance, in the example I give, UUU changes to UUA, and then so instead of Phe, you incorporate Lu, and that may or may not matter. It depends on the protein, and it depends where along the protein. And finally, a silent mutation, if UUU changes to UUC, it's still the same amino acid. Technically, it shouldn't matter unless that same change affects something else that's important on the RNA, and we'll come back to that. So that's the state of knowledge, let's say, in the 70s, and then a big surprise came along, and that is that it turns out that most of our genes don't have continuous segments that code for protein, but rather they are separated by non-coding segments. So you have alternating coding and non-coding segments, and those were eventually named exons and introns. And so the precursor to the messenger RNA has the same information as the gene does. But then we need a process called RNA splicing, which does the cutting and pasting to remove those introns, the non-coding portions. And now we finally have a mature messenger RNA that can be exported for translation. So nobody was expecting this. It seems kind of why, and it's complicated. What's the point of this? And so there's a machine in the nucleus called the spliceosome, splicing body. It's very complicated. It's made up of more than 170 proteins and 5 small RNAs. And its mission is to locate all the exon-intron borders on RNAs made from the thousands of genes and do the cutting and pasting in an exquisitely precise manner. So the scientists who discovered that genes are split into exons and introns got the Nobel Prize also 16 years after the discovery. And I just have here the titles from the 2 original papers from Phil Sharp and Rich Roberts. And it's remarkable that in this title the word amazing appears, because you never see that in a scientific paper. So you can rarely get away with that. Okay, so I've realized this, if you haven't studied this before or it's been a long time, it may be a little complicated, so I'd like to use analogies, and here we go. So we can think of the genome as a library, clearly a beautiful library. This is a real library. It's the Iowa State Capitol Law Library in Des Moines, which I chose because of the beautiful double helical staircase. So each book here represents a gene. So there are 23,000 books in this library. And here's one particular book. We can think of each book as a recipe book or an instruction manual. And this is the one to make a protein called SMN, which stands for survival of motor neurons. So it's a protein that's crucial for nerve cells called motor neurons to be healthy. And so we know the language in which the different chapters in the book are written. It's the language, what I showed you in that table, right? But the problem is that the chapters are separated by pages written in a different language, the language of introns. And so what the splicing machinery has to do is to remove all those extra pages between chapters, and now we can follow the recipe because you're reading the chapters in the correct sequence. And as you'll see, the problem in spinal muscular atrophy is that the spliceosome erroneously removes chapter 7 out of 8 in doing that splicing. And so now the recipe doesn't really work well. And so the goal of the therapeutic we developed is to remind the splicing machinery not to skip chapter 7. Okay, so now I wish to define antisense oligonucleotides, or ASOs for short. So let's look again at the structure of the double helical structure of DNA. Here I've just flattened it, but I remind you, if you look at the sequence of one strand, you can predict exactly the sequence of the other strand. And when this is transcribed, it makes RNA, and that RNA has the same information or equivalent information to one of the 2 strands. So this is called the sense strand, and this would be the antisense strand. That's just nomenclature. So an ASO is a synthetic nucleic acid, so it's similar to RNA and DNA. It differs in some ways, but it's just a very short strand, and we can synthesize it in a machine, and we can dictate what sequence to put in that antisense oligonucleotide. And so that's going to allow it to form base pairs with a particular mRNA that we're interested in, its target. And so the forces that allow this pairing to occur are the same forces that hold the 2 strands of DNA together. When cells divide, the strands separate, and each strand is used to make a copy of its complementary strand. And so, in fact, if you take DNA fragments in solution and you heat, the strands will separate, and then if you cool it, they will find each other because of this, the base pairing. So in the end, an ASO is going to form a helix with the target mRNA, except the only difference is that one of the strands is very short. Okay, so these are short, synthetic nucleic acids. They can bind to RNA by base pairing, And some of them will be able to modify how proteins are made from the target gene as a result of that. And they have chemical modifications to make them more drug-like, that is, to stabilize them, to make sure they're not toxic, to facilitate their entry into cells. And the most important thing is that the sequence of an ASO determines what its RNA target is, and also exactly where along the sequence it will bind in a predictable manner. And so what we're interested in is ASOs that can bind to a target precursor messenger RNA in the nucleus in such a way that it can alter the way it undergoes splicing, and hopefully we can use that to our advantage. Okay, so with that, I can now introduce SMA, spinal muscular atrophy. So it's a neuromuscular genetic disorder, mainly pediatric, and it's a genetic disease that's inherited in a recessive manner. And so what that means is that the two parents are carriers for the disease but don't suffer from it, but they can transmit the defect, the genetic defect, to their children, and 1/4 on average of their children would have the disease. Half of their children on average would themselves be carriers, and one lucky one would be unaffected. Other genetic diseases are dominant, and in that case, even if one parent carries the gene, he has the disease, he has a 50% chance of transmitting that to his or her child. So SMA affects about 1 in 10,000 newborns, which made it the leading genetic cause of infant mortality. So it's a serious disease. And what happens in SMA is that motor neurons in the spinal cord and the lower part of the brainstem undergo degeneration. It's a progressive neurodegenerative condition. And it was known for over 100 years, but finally the defective gene was identified in 1995, and it was named SMN1 for survival of motor neuron, and 1 because there's another gene, SMN2, which is very similar and is unique to humans. And so patients with SMA lack functional copies of the SMN1 gene, but they depend on SMN2. And SMN2 can only express or produce small amounts of functional SMN protein. So that's going to be the target of the therapies that we and others wish to develop. And another characteristic of SMA is that there are different types with varying degrees of severity, and that largely depends on how many copies of SMN2 are present. So the normal number of copies would be 2, and in the absence of SMN1, that means very severe early onset disease. Some people have extra copies, 3, 4, all the way to 8. And so then, even though they still cannot make normal levels of SMN protein, they make more, and hence the disease is milder. And so I'm going to focus today on type 1 SMA, which would be like the two patients on the left, who clearly the disease onset is very early and before 6 months of age. And so it's infant onset, and the clinical observation is that these patients could never achieve independent sitting as a developmental milestone because of muscle weakness and atrophy. And they start out very weak, and muscles get worse over time, and this is extremely sad, but the natural history studies show that they seldom live beyond 1 or 2 years, and if they did, they needed respiratory help, like ventilation, more than 16 hours a day. And on average, that to get to one of these endpoints would require only 10.5 months. So if we compare the SMN1 and SMN2 genes, they're nearly identical. They have just a few differences in those building blocks, a few nucleotide differences. And these studies in 1999, one of which is from here, from OSU, from Arthur Burgess's lab, showed that the key difference between SMN1 and SMN2 is this, what's a C here at position 6 of exon 7. And I should explain, it's a convention, RNA is RNA, but we just show the exons, the coding segments, as these rectangles, the introns as lines, and this shows what is joined to what in the process of splicing. So the 6th nucleotide into exon 7 is a C in SMN1, and a T in SMN2. And if you remember the codon usage table, I don't expect that you do, but the TTC and TTT are synonymous. They code for the same amino acid. So in theory, this shouldn't matter, but it does because it alters the splicing. So exon 7 is always spliced in or included during splicing for RNAs made from SMN1, and most of the time it is skipped over for SMN2. And what happens if it's skipped? Well, you see the normal stop codon is here in exon 7, so the ribosome translates and it stops right here. That's the end of the protein. But in SMN2, that's missing, and so it goes to the next stop codon present in the sequence, so this bit is going to be present at the right end of the protein made from SMN2. So just to show that in a bit more detail, SMN2 produces 2 different mRNAs. That's something we call alternative splicing. The major product is this one that lacks exon 7. The minor one includes exon 7. So when this is translated, it makes this is just a diagram, but it's meant to denote the protein and different parts of it that are important. This is functional. This one is almost the same, but at the end is different, and this version of the protein degrades rapidly. So most of the mRNA that results from splicing is used to make a useless protein. This is crucial, but not enough of it is present. So what we aim to do is to change the splicing so that this becomes the more abundant mRNA. So how do we do that? So this required understanding some basics about how the splicing machinery recognizes exons and introns, and how alternative splicing occurs and is regulated, and how ASOs function. So the first thing we were able to determine is that the reason exon 7 in SMN1 is used efficiently is because there's an RNA binding protein. It's called SRSF1. It recognizes a short string of nucleotides that comprise that C at the 6th position of the exon. So if that C is present along with the surrounding sequence, it'll bind here, and it helps the splicing machinery locate the borders between the introns and the exon. And so the result of that is splicing that includes all the exons, including exon 7. In SMN2, that C is a U, and so now SRSF1 is unable to bind there. And then another RNA binding protein called HNRMPA1 functions as a splicing repressor. It's bound here. It doesn't matter when SRSF1 is present, but in this case, it's no longer present. And so HNRMPA1 hides the whole region from the splicing machinery. And so the predominant product of splicing is going to be 6 to 8, skipping over exon 7. So Spinraza, whose kind of a general name is nusinersen, so I use those two interchangeably, has a sequence that allows it to bind right there, and so it blocks the binding site for HNRMPA1, which is the repressor. And it binds to this sequence that's only present here. It's not present in any other RNAs, at least as a perfect match. And so it's a highly specific drug. It occupies this binding site, prevents the repressor from binding. The other protein is still unable to bind, but the net result of that is the spliceosome can once again locate exon 7 and include it there. And so this just shows you what the molecule Spinraza actually looks like. These are details about its nucleotides. Remember I said that ASOs have chemical modifications, and these are the 3 types of chemical modifications that are present in it. So the reason it's not RNA is that it has these regions shown in, these modifications shown in orange, blue, and green. They all serve important purposes, but they make this very stable and drug-like. So how do we get this to the right place? So it turns out that many drugs are unable to cross what we call the blood-brain barrier. So if you inject them in a vein, for example, they will be around the body except in the brain and spinal cord. And so it's important for a drug like this to be delivered directly to the central nervous system. And so we have something called cerebrospinal fluid that bathes the spine and the brain. So if we put the drug there, then the circulation of the CSF allows the drug to distribute throughout the spinal cord and the brain. And we can do this in mice, and we can inject into this region of the brain that's filled with cerebrospinal fluid, and then the CSF circulation takes care of the rest. It distributes the drug. And then the nerve cells in the central nervous system are able to take up the ASO because of those chemical modifications that I mentioned. So what we're doing here, and let me back up a little bit, these are transgenic mice. The most widely used SMA mouse strain was made here in the Burgess lab. And so why do we need these special mice? So mice have only one SMN gene, which is equivalent to SMN1. And so if we want to fix splicing of SMN2 in an animal, we have to introduce the human gene into it. And also, ASOs being so specific, if you look, even though animal cells contain many of the same genes we do, they're not 100% identical. They have a few differences, and we want ASOs to be exactly able to bind to the transcripts from the human gene. And so that requires these special strains of mice. In any case, so I'm only going to show one data slide, which is this one. This is RT-PCR, so it's a polymerase chain reaction. It's a way to analyze the RNA. Sometimes PCR is used to detect the COVID virus, for example. And so these are samples of spinal cord RNA from mice that were treated with Spinraza, just one injection when they were born, or treated with saline, and Spinraza is dissolved in saline. And then we use RT-PCR to detect these two different RNAs. One represents exon 7 being included, the top band, and one exon 7 being skipped, the bottom band. So these are control mice, 3 of them. You can see they have the right product, but it's not the majority product. It's less than 15% of the total. But when Spinraza is present, this is 6 days after injection, the major band is the top one. So we've effectively switched splicing from being skipped, exon 7 being skipped, to it being included. And this is another assay that allows us to look at the protein that's translated from that messenger RNA. It's called the Western blot. It uses an antibody. And we can detect that protein in the controlled mice, but there's clearly more of it in the Spinraza-treated mice, which is what we want. And the result of that is that the mice live longer and they move around instead of being essentially paralyzed. And so this is a motor performance test for mice. This mouse that just fell is a healthy mouse, but it's not easy. This bar is rotating, and then it's going to change directions too. But this is a SMA mouse that without treatment would have never even been able to move, or barely so, and only lived for 10 days or so. And now it's a 90-day-old mouse because it's received one injection of Spinraza, and clearly it's strong and coordinated. So these results are really encouraging. And so, so this work, which is a preclinical aspect of development of Spinraza, was done in my lab in collaboration with Ionis Pharmaceuticals, a biotech company in San Diego. And so what we did is just what I showed you, experiments in the test tube, in cultured cells, or in mouse models. And once we had this drug by 2008 or so, then Ionis decided to pursue the clinical development. This requires big investments, and it led to clinical trials. But before that, extensive toxicology studies have to be done, both in rodents and in monkeys, to ensure that the drug is safe before it can be given to a patient. And so these are different stages of clinical studies. In the early phase 1 studies, it's just to make sure the drug is safe in a patient. A few patients are tested with low doses. Then by phase 2, maybe more patients, and they start receiving higher doses and multiple doses. And finally, phase 3 is a highly controlled study with many patients, and some of them receive the drug, some of them receive a placebo or a sham injection, and it's in a blinded manner, and it's randomized, well controlled. And so Ionis partnered with Biogen to sponsor the phase 2 and phase 3 clinical trials. And that led ultimately to approval at the end of 2016. And so how is this drug given to patients? It's equivalent to what I showed you for mice, but in order to reach cerebrospinal fluid, the easiest way is to do a lumbar puncture. So at the base of the spinal cord, it's given to the cerebrospinal fluid that surrounds the spine. And one important feature is that because of its chemical modifications, Spinraza is really stable in the central nervous system. Its effects last for months. And so in practice, patients only need to receive these lumbar punctures 3 times a year. At the beginning, they get 4 injections in the first 2 months. Those are called loading doses so that there's enough drug as quickly as possible to start working. But the maintenance dosage is only 3 times a year, and the current dose is 12 milligrams for each injection. So this is a motor neuron. They have a cell body and then the long projection, which is called an axon that innervates muscle fibers. And so the cell body is located in the spinal cord in this region, which is called the ventral horn. And so this is a section of the spinal cord, like going across here. And then, but the axons go all the way out. For instance, this could be a muscle on your calf or your foot. So those axons are extremely long. But what we care about is to get the drug to the nucleus of these cells. And so that's why we have to make sure they get to the spinal cord, and we can achieve that by this method. So to say something about the clinical trials, they began in November of 2011, and they enrolled about more than 300 SMA patients, including infants, children, and teenagers. And those who have remained using the drug since then have been taking it for up to 11 years. The infant trial started a couple of years later, so up to 9 years. The phase 3 trials demonstrated statistically significant and clinically meaningful improvements in motor function measurements, also in survival. And at the time, since there was no other treatment, we didn't know what to expect. And so it seemed possible that you can stabilize the disease, prevent further degeneration. But since we are born with all the motor neurons we will have for the rest of our life, perhaps that's it, But in practice, it's possible to seek motor improvements that continue over time, and that implies that the remaining motor neurons are healthier and they can function better. So I'm going to show a video of a patient, and so this is a patient who participated in the phase 2 trial. He was diagnosed with type 1 SMA at 5 weeks of age, which is pretty early, and given a terrible prognosis. So the parents were told that he would only live for 6 months or so. But they persisted, and they found they were able to enroll him in the clinical trial 2 weeks later at 7 weeks, which is when this video begins. So you'll see that he can barely move, and he breathes abdominally because of weakness in the muscles. He cannot hold his head.
Mr. Cameron is now 7 weeks old.
But so at 7 weeks, he started getting the first dose, and you're going to see how he improves over time. So now he can lift his arms, and he can move his head a little bit. This is at 4.5 months.
You got it.
So he has some hand strength.
You did it. Oh, so proud of you, little man.
Now he's 9 months, and he can keep his arms elevated and reach. Then he turned 1.
Are you working out?
Hi. Are you bouncing? Yeah, you're doing such a good job.
Look at that big boy.
Can you let go?
So at 20 months, he could do some
Do a
stepping.
shot.
Okay. time. Hold yourself up.
Come and get it.
2 years old.
Roll the baby. Roll the baby. Oh, what a strong boy. You gonna go get that train? What is that? Hey.
So at this age, he was using a power wheelchair that can be controlled with one hand, but he no longer needs that.
Push yourself.
A little bit later, he did this.
Push, push, push. Push, push, push. Yeah, keep going. Push, push, push. He is riding a freaking bicycle. So take that, SMA.
Oh my God.
Okay, so that was Dad, obviously very proud. So Cameron is actually going to turn 11 next month. And so remember that before these treatments were available, type 1 SMA meant inability to sit independently. But, so he uses a wheelchair like this nowadays, and he can sit independently, even on his dad. So looking at the patients in the phase 3 study, he was part of a phase 2 study. This is a survival curve, so it means either survival or the need for more than 16 hours a day ventilation. And so the treatment group is in blue, so it's not perfect. We would like to see a flat line. And the control group is there. But the important thing is that there's a big difference. So clearly, it's helping many patients live longer or continue to breathe normally. And so this is a trial that involved 121 type 1 SMA patients. In terms of their motor function, this is clinical data. So this is a motor function score over time. And so there were 40 kids that initially received the sham injection. So they declined slowly over time, and that's what's expected from the natural history of SMA. But the treatment group improves over time, and there are some bumps, but so it doesn't just stay flat. I mean, there's continued improvement over time. And it became apparent, maybe not surprising, that the patients who were treated earlier were doing better. And so then a trial was done where a group of patients was treated after genetic diagnosis, but before the onset of symptoms. And so most of those patients had older siblings with the disease, and that's why they were tested. And that's what was observed. So the slope of this is much greater. So that means that they're achieving good motor function scores much earlier, and this now closely resembles normal infant motor development. So because of these and other measurements, et cetera, so the drug was rapidly approved, and it became the first approved medicine for spinal muscular atrophy. More generally, it was the first approved drug that works by correcting a defect in RNA splicing. Also the first approved nucleic acid therapeutic. There are other classes like mRNA and siRNA. But this is the first nucleic acid therapeutic for neurology. And that trial, called the NURTURE trial, that showed that if you treat pre-symptomatically, you can prevent the onset of a neurodegenerative disease, in this case SMA, that had not been done before for any neurodegenerative disease. Disease modifying refers to a drug that gets at the root cause of a disease and it changes its course for the better, and so that's a disease modifying drug. And so the drug itself was recognized by this very important prize in pharmacology. It's called the Prix Galien. So it received a silver medal as best orphan or rare disease drug in a period of 50 years from 1970 to 2020. And so let me give you some updates. So this is, I want you to pay attention to this video. This kid has type 1 SMA, genetically diagnosed, and the difference from the previous one is that he was treated at 9 days of age thanks to newborn screening. And so in this video, he's 16 months old and he's learning to walk, And you would never guess in a million years that he has SMA of any type. And so clearly, early treatment, pre-symptomatic, is really should be the goal. And so, fortunately, in the U.S., now all newborns are tested for SMA. There are also two other approved therapies. One is a gene therapy that was developed in part at Nationwide Children's Hospital. And another one is a small molecule. And so patients now have these options, but crucially, they should be implemented as early as possible. And so more than 14,000 patients have been treated with Spinraza in over 60 countries. So we'll let Franco finish walking. It's really amazing to see this. He's from Argentina. He's almost done. There we go. Okay. I wouldn't interrupt this. Okay. So there's still more work to do because patients that have more advanced disease, it would require some sort of regenerative medicine to restore better function. So our job is not done. And so there are ongoing clinical trials employing Spinraza, for instance, in combination with other drugs or molecules that are similar to Spinraza. For instance, two of these trials are utilizing different drugs in combination, and those target a protein that's very important for muscles. So the Spinraza is acting on the motor neuron side of the neuromuscular junction, the union between the nerves, the motor neurons, and the muscle fibers. And these two other drugs would act on the muscle side by inhibiting a protein called myostatin. And so one of these already got very encouraging results showing that the drug combination works well. So they should get approval very soon. There's another trial that involves higher doses of Spinraza. Instead of 12 milligrams, they want to see if they can get greater efficacy given 50 and 28 milligram doses. That also, they just announced that they're getting positive results. So that may become the standard dose. I'm not sure yet. There's another trial that involves a different way of giving the drug that maybe would be more convenient. So this involves a port and then a catheter system to reach cerebrospinal fluid. And then the last one is a new generation of chemical modifications to make a version of Spinraza-like drug that maybe only needs to be given every 9 months instead of every 4 months if it works. So there have been 11 drugs of this type approved to date. So I told you about nusinersen or Spinraza, and then one of these is no longer used, but there's another 10 drugs that are used for different diseases, like Duchenne muscular dystrophy and several others that are mentioned there. And so several of them act by modifying splicing, but the others have a different mechanism, and I'll just mention that very quickly because it justifies my title better, shooting the messenger. And so those are called gapmers. So this is nusinersen or Spinraza, and this is a different drug for Duchenne muscular dystrophy. A gapmer has wings that have the same modifications as shown here, but in the center they have DNA nucleotides, deoxynucleotides. And the reason for that is when this pairs with RNA, if you have a DNA RNA helix, there's an enzyme in the cell called RNase that will recognize that, and it will cleave the RNA strand. So effectively, that destroys the target RNA. So why do you want to do that? You wouldn't do it for SMA, but you can do it for instance, a dominant genetic disease where the defective protein is toxic. It's causing toxicity. And so, for example, some of these are used for ALS or are being developed for ALS, for Huntington's, et cetera. So, the ones that modified splicing, there are 5, but the other 4 are for the same disease, Duchenne muscular dystrophy. So I'll explain how one of them works. I showed you that Spinraza promotes inclusion of an exon. So this drug, called eteplirsen from Sarepta, promotes skipping of an exon. And actually it has different chemistry shown here. So that's something you want to do in a special case. So the protein that's defective in Duchenne muscular dystrophy is called dystrophin. It's a huge protein. The gene is like 2 million base pairs. And so the protein is not functional if it's truncated. And so, for instance, there are deletions of the gene that remove some of the exons. And so the protein that's made from that is what we call out of frame. The ribosome is reading it, and then it gets to, it's because exons are missing, the next exon is out of frame. And so it quickly gets to a stop codon, and so this cannot make functional protein. So if using an ASO to skip one more exon, then these other exons restore the reading frame. So the reason is that the words are read in sequence of 3 base pairs at a time, 3 nucleotides at a time, and so the exon borders can be before a word or in the middle of a word, and so if an exon that's missing is not a multiple of 3 nucleotides in length, everything after that is read in the wrong sequence. So the dystrophin gene has 79 exons, and here I think we're using a drug to skip exon 51. But it allows to make a protein that's missing a portion in the middle, but it has the ends. And for this particular protein, the ends are all important because they anchor 2 different structures in the muscle cell. And so, in effect, there's another disease called Becker's muscular dystrophy, which is milder, and the protein that's made after those mutations looks like this. So this therapy is turning a more severe disease, Duchenne, into a somewhat milder one, Becker's. And so the other drugs target different exons. So this is like a version of personalized medicine, because for each Duchenne patient, you would have to know what's the mutation and which exons would you have to skip in order to be able to make some protein that at least has some function. So there's 4 different drugs targeting 3 different exons out of 79 exons. And so another example, and I'm about to finish, but this is from a company I co-founded called Stoke Therapeutics. And so they have drug development for these various conditions. The most advanced one is for something called Dravet syndrome, which is a genetic form of very severe epilepsy. And that's a dominant disease. It's a kind of dominant disease called haploinsufficiency, which means that you have one healthy copy of the gene and a defective one. The defective one makes a protein that's inert, it's not functional. The healthy one is there, it makes protein, but it's not enough because it's only 50%, half of what you really need for normal function in for this particular protein. It's a channel that transports sodium ions. So it turns out that there are some genes that naturally have alternative splicing that includes what we call poison exons. They're there maybe for regulatory purposes. So if that exon is included, it does not make protein. It's not productive. And so if it's treated with an ASO to skip that exon, so similar to what the Duchenne drugs, but here we're removing a bad exon, then what we could do is make more of the protein from the single healthy gene, healthy copy of the gene. So let's say we make twice as much, it's basically equivalent to the normal state. And so this drug has completed phase 2 trials, and phase 3 trials will start very soon. The results are very encouraging. So these are just examples. There's a lot of ongoing work in industry and in academia to develop drugs for cystic fibrosis, Huntington's. We're working on a pediatric form of brain cancer that's very deadly, and I'll talk about tomorrow at a seminar here. And but that's so. I think there will be lots of approved ASO drugs. That's my prediction. And so with that, I just want to acknowledge people in my lab who worked specifically on SMA, and especially Yemen Hua, who was a postdoctoral fellow at the time. He did much of that work. And we collaborated with Ionis Pharmaceuticals, where Frank Bennett and Frank Rigo were our closest collaborators. Biogen also obviously played a role, as did many clinical trial teams around the world. And most of all, I'm grateful to the SMA patients who participated in the clinical trials and their families, and then the foundations and our sources of support. Thank you, and I'd love to take any questions you may have.
Okay, so what we'll do is we'll have some open question time for Dr. Krainer, and then we'll have a chance to go out to the reception and see the students' work and also talk to him one-on-one. And we have 3 people with microphones, and I will go ahead and put your hand up. Go ahead, Karine. Yeah.
Hi, thank you. It was a wonderful talk. I'm Sumi Vasu from the College of Medicine at Ohio State. It was very heartwarming to see the progress in translation for these young kids. There are many, many debilitating pediatric rare diseases. How can you predict which one is amenable to ASOs? Do they have to be as defective splicing, or is there a mechanism to predict?
Okay, yeah, great question. So nowadays, whole genome sequencing is increasingly being done. The cost of doing that is less, and it can be done rapidly. So locating the defective gene and understanding from the sequence what's the lesion or the potential lesion, we cannot always predict or interpret robustly, but in some cases it's obvious that it's a problem with splicing, or it's a problem that might be corrected by altering splicing in some way. And then there's the other version, the RNA-seq version, which is to remove the protein or reduce its levels. And I didn't get into it, but splicing, because it's so pervasive, so we have about 200,000 introns in our 23,000 genes and lots of alternative splicing. So if we are able to manipulate that, so we can change the product that's made. Sometimes we can increase how much is made, as in the Dravet case. And other times we can reduce the concentration if it's toxic. So it can be used in many different ways. So I'm not going to say that ASOs can be used for everything, but there are, I think, many opportunities. You're welcome.
There's another one.
Like the child in Argentina.
Where
Will
are you?
they be
Sorry.
treated for the rest of their lives?
Can you raise your hand? Oh, yeah. Okay, thank you. Yes, so that's what we think, that there's a continued requirement for SMN. So some of them have been treated for 11 years, so they've received more than 30 lumbar punctures. It's well tolerated, and of course they have other options. The other drugs are given in a different way. I can get into that if you want. But we believe that that they'll need it for the rest of their lives. Yeah.
Go ahead, Karine.
Nice talk. So as you know, the goal of ASO try to rescue the functional protein. So as you know, NC is also developed AV-based delivery of functional protein in vivo. So I'm curious, what's the advantage of ASO over the functional AV delivery? Thank you.
Okay. So one of the therapies that I mentioned involves using a virus to introduce a version of the SMN protein. It's called a cDNA. So it's like the gene minus the introns. And so the virus is introduced, it infects most or many of the motor neurons, and it forces them to start making the correct protein. So in theory, that requires one treatment. So the issue, and so that's an approved therapy for patients under 2. And at the moment, it's being tested also for older patients. So each of the methods or therapies has pros and cons. I mean, I think there's an argument to be made that you may need both. For instance, the gene therapy may take a little bit of time to start for the virus to get in and start expressing the protein, and so you may have to take one of the other drugs as a bridging therapy so that you can get the benefit right away, but then maybe you don't need it forever. But theoretically, the virus may stay there and produce a protein for the rest of your life, but we don't know that, right? I mean, we can follow that and see if is it true after 10 years or not, or do you need to then still complement that with one of the other therapies. And then the other issue is how to get all the motor neurons to take up the virus, because if it's only, let's say, 70%, then you're not getting the benefit of the other 30%, and you're going to lose those motor neurons. So those are arguments for maybe combining therapies. The problem is that all the therapies currently are very expensive. So I don't know. I mean, I think ASOs have some benefits. It's not a permanent change. They don't. There's not an immunity issue. So if you get a gene therapy, you won't be able to get it again, or to get a different gene therapy, because there will be antibodies to the virus. So you have to consider all these issues.
I was just wondering, this is primarily benefiting neuromuscular diseases. Could you explain how it works with Huntington's chorea?
Right. Yeah. So there isn't yet an approved ASO for it. So it went. So several companies have worked on it. There have been phase 3 trials that were not successful, but they haven't given up. So in Huntington's, one of the genes has an expansion, which is toxic. So it's an example of a dominant disease. So the idea there is to destroy the RNA from the gene that has the expanded repeats. And so there's some difficulty selectively destroying the RNA made from the mutant gene versus the normal gene. And so some experiments in mice suggest that maybe even reducing both is probably better than, it's maybe not ideal. And so they're trying to come up with ways to selectively reduce the expression of the mutant allele, not the other. The methods tried to date didn't distinguish that, or at least one of the trials didn't. So why didn't it work? Well, we don't know. I mean, it could be that the drugs were not potent enough, but it may be the nature of the disease, right? For a disease for which there's no therapy available, there's nothing to compare it to. So you're not quite sure what group of patients should you test. Maybe it was too late. Maybe they needed to be treated earlier in the disease progression. Maybe the trials didn't last long enough. It could take 2 years before you see what you want to see. So there's all these unknowns, and we face them also with SMA. Luckily, it worked out. For ALS, there's one form of ALS where a drug was conditionally approved, and it also failed the initial clinical trials. But then they did it again, and they waited longer, and they began to see a benefit. So that's where it is. I mean, I think potentially this may work for Huntington's, but we don't know yet.
Okay, Amrit, go ahead. This will be the last question in the public portion. Go ahead.
Okay. Hello. So I know that you mentioned the chemical modifications for ASOs. I'm assuming that's for to protect the ASOs from nuclease degradation.
Yeah.
What is the other functions of adding the chemical modifications?
Okay. Yeah. Great question. So you're right. I mean, RNA is very labile. So the modification that's shown, that it's called 2' MOE, that protects from nucleases, because the nucleases attack that position to destroy the RNA, and with that chemical modification, it blocks. It's like very, very robust. Then there's another modification in the backbone, in the phosphate backbone, and that improves or increases binding to proteins. And so when an ASO is injected, what can happen? It can be taken up by cells, or it can go through the kidneys and be excreted in the urine. And so binding to proteins like albumin in the body means that it stays there longer, it has a chance to be taken up by cells. And then it also binds to proteins on the cell surface, and they help internalize the oligo. And then the last modification is a methyl group that's put in the Cs, and it blocks innate immunity. So if I know last year there was a presentation about mRNA vaccines, and they have pseudouridine instead of uridine to block immune responses, because the cells, when you inject you deliver a nucleic acid to them, they think it's a virus, and so they mount. There's various defense mechanisms. And so oligos would be toxic, but for that methylation of the Cs, and then it's not, it blocks this immune response.
Okay, thank you.
You're welcome.
Okay, let's thank Dr. Krainer for a wonderful talk.
Thank you.
We're going to have just some quick closing remarks.
So if you don't recognize me again, I'm Dan Schoenberg, name of the lecture. When I was asked last year who I thought would be a great speaker for this series, The first name on my list was Adrian Krainer. So Adrian, thank you again for coming. Please join me in thanking Adrian. So, quick question. How many of you have had either a Pfizer or a Moderna COVID vaccine? Show of hands. Okay. So that's a good example of RNA biology. And some of the work that's been done to develop that came out of Ohio State. So that was done by our trainees. I encourage you to go see the posters, talk to them, see the kind of work that they're doing, and support this kind of work going in the future.
On behalf of Science Sundays and in our partnership with the Center for RNA Biology, we'd like to thank everybody for coming. Please join us for a reception where many of the students will be presenting their work in this area. You'll have a chance to ask questions of those students, have a cookie, have a coffee, meet Dr. Krainer. It's all great. Thanks very much.